Hereby, I open this academic ceremony in which Camilla Soragni will defend the academic thesis, Next Stop, Screening on a Chip where Biology Meets Scalability, Development of Assays for Placenta-on-a-Chip Models. Dear candidate, may I invite you to present a summary of your study and conclusions of your thesis.
Thanks, Director. Dear Director, dear member of Corona on site and online, dear colleagues, family and friends, thanks for joining me here today and welcome to the presentation of my PhD thesis entitled Next Stop, Screening on a Chip where Biology Meets Scalability, Development of Assay for Placenta-on-a-Chip Models. I would like to start by introducing the placenta, the temporary organ which develops over pregnancy. It is an interface between the mother and the fetus, and if you look at the structure, it looks a little bit like this. On one side, we have in red and blue the vein and arteries of the mother, so the vasculature. Then in yellow, we have the main component of the placenta, the trophoblast. And on the other side, again, in red and blue, the vasculature of the baby. And we need to study the placenta to develop assay because there's little knowledge about this organ, and this is due to the fact that it's very difficult to study directly in human, of course, because there's a pregnancy, so it's a sensitive time, and there's some limitation by studying in animal because they generally present a placenta with a different structure, so then it's difficult to use this data back in human. And then, so we need to strongly rely on cell-based model. And because of this, we need that this cell-based model are close as possible to the human, so in vivo situation. But the traditional way to culture cell is as one layer of cells on top of which we put media with nutrients. But if we look at the vein that we were discussing before, we have this tubular structure inside of which there's the blood which flow. So it's quite different of what I just described with cell culture. Luckily, we have this new advanced cell culture technique called organ-on-a-chip, which allows to culture cell in a 3D structure. As you can see in the sketch, is a kind of tubule, inside of which we can perfuse the media. And of course, this is closer to the human in vivo situation. In this project, in particular, we use a platform called the organoplate, which contains multiple chips. And as a model, we use a model of the placenta which recapitulates the placenta-fetal interface. So if we zoom to the structure, we will have on one side in yellow the trophoblast cells, and on the other side in red, to mimic the fetal vasculature, the endothelial cells from the umbilical vein. The model, the cells, the trophoblast cells were exposed to a syncytialized media to stimulate syncytialization, which is the process to mature the trophoblast. So until now we talk about the placenta, and I would like to start now describing what is an assay and talking about the assays developed. To introduce the concept of an assay, I will make use of a very simple test, but that I think that everyone in this room has tried at least one time. I'm talking about the COVID test. So we all know that we need to collect the sample, wait to run the test, and then we really hope to see just one line. This line next to the C, which stands for controls, which indicate that the assay has worked and we don't have COVID. We don't hope to have another line because that means that the analyte has been detected and so we are positive to COVID. Because the assay is a procedure to detect an analyte. So for this project, we didn't develop an assay for COVID, but an assay for ROS. What are those ROS? ROS stands for reactive oxygen species, which are a class of molecules which contain oxygen and are highly reactive. Because those molecules are produced every time that we use oxygen, and because they are highly reactive, they can react with all the molecules in our cells and body by damaging them. To avoid this process, there is a class of molecules called antioxidant defense, which can react with this ROS in a controlled way and eventually produce water. But in case this ROS, the quantity of ROS produced, is higher than the antioxidant defense, we will encounter a situation called oxidative stress, which is generally linked with inflammation and disease, and in case is too severe, would lead to cells apoptosis, so cells death. But ROS are not just harmful molecules, they are also, they play a role as signaling molecules, so the presence of this molecule will trigger physiological processes. And this is the case for the placenta, because at early stage of the placenta development, a lot of angiogenesis take place. Angiogenesis is the process of developing new vasculature from a pre-existing one, and this process requires also the presence of ROS. So in order to have a healthy placenta development, we need ROS. Moreover, the trophoblast cells, every time that make use of oxygen, they produce more ROS comparing to the majority of cells. Next to that, at early stage of the development, trophoblasts they don't express antioxidant defense, which increase over time, but all over the pregnancy we have a slow amount of oxidative stress. And this is necessary. Anyhow, again, if the oxidative stress is too high, we will encounter pathological development of the placenta, and so a disease. So the assay that we developed was to quantify ROS and cell viability. Cell viability is a percentage between the total number of cells and the dead cells. The assay is pretty simple. We just incubate the culture with a compound, and this compound, if the analyte is present, will give a fluorescent signal. We use green, the compound gives a green signal for the ROS, a blue signal for the total amount of cells, and a red signal for the dead cells. As control, we use a strong oxidant called terbutyl hydroperoxide, which, as you can see from the picture, gives a very strong green signal, so means that the ROS are present without killing the cells. We apply the assay at our standard culture conditions, so when the trophoblasts were syncytialized. We can see that the control has worked, as in both types of cell we have this strong green signal. And when we look at our sample, so the model, we can nicely see that on the site of the trophoblast, we have a weak, but we have a weak signal. And this is what we expected. We also check how this syncytialization media will play a role in the way that the cell experience ROS. But unfortunately, we couldn't see a difference when we didn't expose to the syncytialization media. Anyhow, both the condition, when we look at later time, we could see that there was a decrease in the ROS signal on the trophoblast side. And this is expected because, as I mentioned before, over time we will see an increase in antioxidant defense and so a decrease in ROS. So until now we talk about placenta assay, we didn't tackle yet the screening. So I would like just to give you a definition of what is a screening. It's a large-scale test of compounds. And the reason why we need a screening in the context of the placenta is because the majority of the drug already available in the market is not known about their safety use during pregnancy. So we need to test a large amount of compounds on this placenta-on-a-chip model. But I would like to make use again of the COVID test because, of course, I think if you have to run the COVID test for just one person, or I think to have to learn for a large amount of people, or even bigger. So passing through the assay and the scalable assay require a lot of work. Because organ-on-a-chip technology are generally used in a small, medium scale, because they are quite complex model, and generally complexity and scalability go in opposite direction in science. So as a first attempt, we didn't use a model which was quite complicated as like the placenta-on-a-chip, but you use a kind of simple version, so what is called vasculature-on-a-chip, which is just the endothelial cell, so what was the fetal side in the placenta model. And as assay, we use this angiogenesis assay, which trigger angiogenesis, so the formation of these sprouts. And we tested this large amount of compounds. And we look for compounds which were capable of inhibiting angiogenesis, so basically killing the sprouts, without affecting the main vasculature, so the main tubule. So in this graph, we show the results of this screening. Each dot represents a chip. This is an image of a chip where we have an intact tubule and sprouts. Of course, not these dots are like this, but this is just to give you an idea of the model that each dot resembles. So out of more than 1,500 compounds, we could identify 51 that were active, so they kill the sprouts without killing the tubule. And out of these 51 active compounds, we find 9 that they were already known to be active, so to just inhibit sprouts formation, so angiogenesis. So these results of these 9 compounds that work validate a little bit our screening. The fact that we could find still a lot of compounds that they weren't known to be active, these indicate that these potentially are new drug that could be developed. And but we didn't show just this with this work. We show also a new approach to applying screening, of course, by using this organ-on-a-chip technology, which presents a much complex model comparing to the traditional 2D, and which allow us to check for both efficacy and safety. And safety generally is not checked at this early stage of the development of a drug, while it plays a big role then in the failure of the development of the drug. So integrating this information at this early stage will be very beneficial. And this is due to the fact that we are using a complex model, otherwise it wouldn't be possible. Of course, the next step will be to run a screening with this placenta-on-a-chip, so to test all of these drugs. And more, but if we look more in general, this work, of course, is a validation of this technology, the on-a-chip technology in a large-scale setting. But more in general, with this example, as the first application of a complex model in a large-scale application, we bring scalability a step closer to complexity. And with this, I would like to thank you all for your attention, and I hope to have been clear. Thanks.
Thank you, dear candidate, for your very beautiful and clear presentation. And now the opposition will be opened by Professor Sluimer, and she's Professor of Cardiovascular Pathophysiology at our university. And in addition, she's also chair of your thesis assessment committee. Professor Sluimer.
Thank you, Prorector. Dear candidate, very nice to hear your layman speech. It's the first time I think I encountered this comparison with the COVID test, but I think it's going to be more, but it's very original and very topical. Very nice. I also enjoyed reading your thesis. People always say that, but for me it sort of brought me back a little bit to my own PhD time, where I was working on angiogenesis and atherosclerosis. So I always loved the angiogenic field. It was always a bit of a doubt that I should have gone into the tumor angiogenesis, but anyway, nice enough I can get to read your thesis now as well. So congratulations also on this great collaboration with industry. A nice example how the ITN network, the doctoral networks, can bring both academia and industry together. So well done to you and well done to your promoters team, so supervisory team. But of course, I do have a few questions, although I really like the thesis. And I'll start, I already said I used to work on this endothelial cell biology, and when I was a PhD student, I went to these conferences and the International Vascular Biology meetings, and there was Bill Aird, well, a long time ago now. He presented work where he explained about endothelial cell heterogeneity, and I was really impressed, and this was even before the era of single-cell sequencing. So can you explain this concept of endothelial cell heterogeneity to me and what it would mean in terms of your thesis?
Highly esteemed opponent, thanks for your kind word about the presentation and the thesis, and thanks for your question. As you can see also in this picture here, that we have a representative image of the angiogenesis model that we use at BMate as a company where I developed my PhD. We have two types of endothelial cells in here because the one on the main tubule are what is generally called quiescent endothelial cells. So are endothelial cells that are not in the phase of in angiogenic phase or in the phase of sprouting. While the side of the tubule which face the sprouts in this picture, and so the ECM generally, those are the angiogenic endothelial cells because they has been triggered by a cocktail mix of compound that we put on the other side, on the next chip, next channel that you cannot see here, which create this chemical gradient. Those layer part of the tubule is triggered to start migrate and then produce the tip cells and then the sprouts. And so we have these two different types of endothelial cells. And with the ROS assay, we could a little bit confirm that these sprouts present a higher level of ROS comparing to the main tubule, which was something that is already known. It's very well known that ROS trigger angiogenesis. But yeah, this is a little bit new to properly show a picture with this signal. Yep.
So that's well explained. So that's indeed more the state of the endothelial cell, which indeed also reflects heterogeneity. But what about differences between organs or arterioles and capillaries and venules? And so can you explain a little bit more about this and how you incorporate this in your models?
Yes. So in our model, we use also for like the angiogenesis model, the endothelial cells from the umbilical vein. And the reason why we generally use this kind of cell line already available is because we always have this scalability in mind. And so in order to have a scalable model, you need also easy available source. But of course, like in different organs, like to be more maybe precise in certain kind of model, and especially if we want to go not for scalable model, but more like personalized model or personalized medicine with this free model. Of course, like using the cells of, for example, a capillary rather than a vein will make a difference. And of course, using like primary cells or patient-derived material will be also more beneficial. In this project, because it was really focused on scalability, we just use the simple version. Also, like for the placenta, we use like the BWO, so that are also commercially available. So that was a little bit like...
yeah, so I understand the trade-off between the scalability and what's practical, and you can get a lot of cells out of the placenta, but I really do wonder that there's a lot of different flow patterns and shear stress, and so the gene expression patterns and also the response then to your stimuli and your drugs will be very different. So is there already data out there that has done like single-cell sequencing of all the cells or the endothelial cells of the placenta, so you can compare maybe your UVEX to the real in vivo situation, so to see, yeah, to make the trade-off a little bit more in favor of a true biological phenomenon.
Yes, absolutely. I would like to just specify that the placenta model was developed in parallel with this project. So this was the first step, let's say. Then, of course, because there's the interest of developing also disease model, of course, there a better investigation about which kind of cells, let's say, to use to recapitulate also which kind of disease will be necessary, because that was like the first model establishing this platform, and was also with the main focus of scalability because of this big problem of not available knowledge about the drug, the safety use of drug during pregnancy. That was the main focus.
That's good, and you already mentioned some ways forward, like the using cells from the blood and maybe making iPSC. So that's great. So there's even more issues with heterogeneity, right? It's not only the heterogeneity between the endothelial cells, but also there's a lot of variability between humans. Now, you took that into account a little bit by using a pool, but I'm still a little bit worried, and maybe you can tell me your concern, if you share these concerns or not, that you, with only one pool, and if you would use the next pool, you would get very different results in your screen. So do you have any information on this, or how do you take this into account for your future work?
Yes, well, I think that, well, generally in the screening for screening drugs, that they generally go for even a high throughput, like the standardization of the process is very important. So for this first experiment, for this first screening, we use the vasculature model because it's the most used in the company. Was one of the first that the company came out. So we know quite well the model was already automated. So because the seeding was done with the robot, and so.
But how can you make it better?
Make it better, of course, like using, because the next step will be also to use disease model, because if you have to find a treatment for a disease, it's good to start with the disease model. Like, for example, in this case that we use angiogenesis, that could be a more like, instead of using the UVA, the patient derived material that, or like iPSL, these kind of things, that then, but before there's need of a lot of standardization, I think, to reach that level that we can put in a robot, because we try also like sometimes with other model, maybe that were a little bit more complex, and that the limitation is really when you have to, because for the screening we have like 4,000 chips, and so it's hard to have then the replicability, but of course like using of like iPSLs or these more relevant, biologically relevant cells will be much more beneficial.
Thank you very much. I think I've run out of time. Kind of short question, and maybe not too long an answer. So you already said you wanted to improve the model and reflect more the disease situations, etc. So the other investigator that I was really always very impressed with was Professor Vestweber, and together with his colleagues Jaap van Buel in Amsterdam, they showed that endothelial cells, or immune cells, cannot only migrate between endothelial cells in case of inflammatory settings, but also through the endothelial cells. And you have not taken this inflammatory component into your model as of yet. So I was wondering, is there a role for immune cells in the placenta development at all, or in a disease setting? And would you consider including it?
Yeah, it plays a role in the placenta development, and I agree that it could be nice to include it, but before maybe include that, the next step will be to include also the spiral artery of the mother, because in this model we didn't include yet, and then maybe further, because already with the spiral artery is a tree culture, and then going further maybe with some immune system. But again, like that will require even a harder practical limitation, because
and not easy, but I think it would be more reflecting at least the real life situation with circulating immune cells. I thank you for your answers, and I give the word back to the prorector.
Well, thank you, Professor Sluimer. The opposition will be continued by Professor Moroni. He's Professor of Biofabrication at the Merlin Institute of Regenerative Medicine of our university. He is a member of your thesis assessment committee, and in addition, today he volunteered to be secretary of your promotion committee. Professor Moroni.
Thank you, Prorector. Dear candidate, I also would like to congratulate you and your supervisory team for a very nice piece of work. I enjoyed reading it. Sometimes also flabbergasting, and I think also this last picture, actually, that is also part of your thesis cover, exemplifies the excitement actually that came when reading your thesis. Now, there are a few questions that also have been triggered by reading it, and not necessarily actually on the details of the thesis, but really I would like to reflect with you more on the overall impact and the next future steps of your work. And to maybe initiate a discussion, I would like to ask one of your paranym, if possible, to read the proposition number 5.
On-a-chip technology has reached a mature stage where complex and physiologically relevant biology is combined with scalability, rendering them a valid alternative to in vitro 2D cell culture.
Thank you. So can you elaborate on what do you think is needed to really push this towards daily use? Because we still culture cells onto the cell culture plates, despite, I think, all of us also tell our students that this is very far from what should be done, but we can't get rid of them. Not of the students, but of the 2D cell culture plates, just to be clear.
Hi, esteemed opponent. Thanks for your kind word about my work, and thanks for your question. So I think that, of course, it depends on what we compare between 2D and organ on a chip, because the main difference there is in terms of cost, because it costs more and is not a technology that everyone is familiar with. But it has been shown already, for example, if you look at the endothelial cells model, the vasculature model, that in a 3D structure with perfusion, the cells perform better. They present a morphology that is closer to in vivo and a functionality that is closer to in vivo. So it has been shown that in terms of physiology and biological, from a biological point of view, it improves the traditional cell culture. But again, the main skepticism, people were mainly skeptical about the fact that you generally apply this on-a-chip technology in a very small, small throughput. So a lot of people they're looking for the validation, because once you validate on a high scale level, you are sure that maybe I can invest in this technology because I'm sure that you have hundreds of chips with the same condition that show the same, while before, maybe you can show me like 10 chips were already quite a lot for one condition, and so...
No, I understand, clear. And in view of time, let me interrupt you, because then I'd like to go a bit more into dialogue with you. So clearly, you have now tested 1,500 and more compounds, and if I did the math correctly, 42 were new compounds that could be promising, and 9 were already compounds that actually are known to be new angiogenic compounds. Have you also, do you think it's necessary now to go back to the cell culture and test those 42 compounds to really prove that with your chip technology you have better efficacy, or would that not be needed anymore?
I think that maybe it could be nice to test it, but the problem is that, of course, like we cannot test both efficacy and safety. We cannot make this straightforward comparison, because generally in 2D angiogenesis, like the model of angiogenesis are actually not model of angiogenesis, because it's not the real development of a new vasculature from a pre-existing one, but it's just rather you culture the cell as a monolayer, you scratch, and then you wait, or you, I don't want to diminish the thing, but they generally don't have like a main vasculature from which you
clear.
sprout.
And again, it's not that I'm a fan of 2D culture, so I'm not here defending that, but I'm just trying to understand critically what we can all do together to really get rid of that culture plate that is so obsolete. But there are, for example, some assays where you have endothelial cells covered into a microsphere, and you put it in a gel, and in principle, from there you can indeed see sprouting from the microsphere. Or if you have your UVEX or other endothelial cells on matrigel, you typically would look at tubular formation.
Yes, we generally embed it with the ECM, and they form a tubule, but they don't form a tubule from a pre-existing one, let's say. But I think that it could be nice to compare them, also to validate the fact that yeah, maybe, you know, because we can have different scenarios. We can have that in 2D, we have the same results as we did with efficacy, and we still have the fact that with this model you can check also safety.
Yeah.
Or we can have different results, and so for there, like, of course, like the 9 compounds that are already known because they are known for in vitro or in vivo, like the ones that are known for in vivo, if we could find.
Yeah. And if we now jump now to indeed the other side of the spectrum and look at animal models, do you think that this kind of test would be able to replace existing animal models, or would you always combine it with a final proof in an animal?
So the field of organ-on-a-chip in the drug development field, it fits into part, let's say. Originally, it has meant to replace animal model, but for that we are not talking about organ-on-a-chip, but body-on-a-chip, because then you need the complexity of the animal, because we still rely on animal, because you don't have just the placenta or the angiogenesis, you have the rest, all the other organs, and that is necessary.
Yeah.
And at that level also you don't need to be so scalable, because we're not talking about this
1,500 compounds.
Exactly.
No, clear.
So with this example, actually it fits in the early stage, so it's another, let's say, approach of organ-on-a-chip. But organ-on-a-chip like this, no, cannot be a direct replace.
Okay. I have actually a few other questions on your placenta-on-a-chip and how, if you have thought about chorion or amnion on a chip as well, but I also see the prorector nodding in not the nice way that I wanted, so I'll give back the word to him. I'm satisfied with your words, and thank you.
Thank you, Professor Moroni. The opposition will now be continued by Professor Zacchigna. She's a professor of cardiovascular biology at the University of Trieste, and she also works at the International Center for Genetic Engineering and Biotechnology at Trieste. She's a member of your thesis assessment committee, and our university expresses its gratitude to you, Professor Zacchigna, for taking part in this opposition today. Professor Zacchigna.
Thank you very much. It's my first experience here, and I'm very happy, and I want to congratulate the candidate for her excellent work, and also to all of you for how this defense is conducted. It's very nice to be here. So coming back to your thesis, I saw that it was very technically oriented, so you develop a lot of essays, and your discussion, you provide a nice overview and insights on how the technologies you developed in your work could be used and exploited. So my question is if you have to invest your money in one of the applications in which your technology could be used, what you would bet on? So what do you think it's a more exploitable aspect of the essays you develop? And also, since you are in this transition between industry and academia, can you comment a bit on the industrial, scientific, and clinical impact that this technology could have, and what could be the priority, according to you, for which it could be used to optimize the impact?
For your kind word and for your questions. So, well, I think that because I not work just only with the placenta, but that was I was part of this iPlacenta consortium. So if I will have to put some money, I will put it on bringing the placenta-on-a-chip on like an automation version to run a screening, because I think that we need to test those drugs for like their safety use during pregnancy, because these not only affect, of course, the mother, but affect future generations. So this has a very long-term impact, and it's quite, to be sincere, quite strange that we have a lot of technology, but we still have a little knowledge about, you know, all the pregnancy-related field, which has a very impact, because a lot of disease also they can affect the fetus and so the baby, and so long-term effects. So if I would bid my money, I would do something like this, because I think that will have like the main impact personally. Then in terms of like this technology, how can impact the industry or the clinical aspect? Well, of course, like the first impact is like in the industry, in case, you know, pharmaceutical company will be interested to use this technology to apply new drugs. And of course, the long term, because it takes a lot of years to develop drugs. Of course, if this technology could be implemented in an industry field, then later on we'll also have some effect also on a clinical level with maybe new therapeutic or what I was saying before with the placenta, having the knowledge about what I can prescribe, because going back to the drug and placenta issue, those drugs are still prescribed. It's not that they don't get prescribed, but we don't know what's happened. So,
right. And in addition to drug screening, could you envisage additional screenings, for example, to investigate some molecular mechanism that you deem interesting or needed to understand, for example, pathological condition or physiological development? So something to get insights on the molecular mechanism that involve placental biology.
Yes, I think that could be also used in that sense. Maybe like the screening approach for like the development of maybe a new model and understanding like the mechanism maybe is a little bit let's say reductive, because there's in order to reach scalability maybe we have to give out some resolution, let's say. So for like if we want to test like different compounds and understand the mechanism, let's say, of action of this compound, yes, but maybe to understand the mechanism of like the placenta development and everything, I would personally go more on like less on scalability and more on resolution. So more, for example, on like a more complex model, or rather, as we were discussing before, using primary cells or patient-derived cells. I think that will have more will have more benefit, let's say.
I was thinking at the genetic screens, for example, using siRNA-based screening or microRNA screening. Do you think it could be feasible?
Yes, we don't do a lot of this kind of analysis on the on-a-chip, for one reason, that you need a lot of material. So you need a lot, so that, yes, you need a screening, because you need to pull multiple chips together in order to have enough material to run the analysis. So yes, of course, I was just thinking before about the screening in terms of just drug development, not more in general. But in that case, yes, because it will fit like the large scale amount, and the fact that we can still study this mechanistic development of the placenta, or rather disease. So
great, thank you very much, and congratulations again.
Thank you, Professor Soragni. The opposition will now be continued, I should say, by Professor Biesen. He's Professor of Experimental Vascular Pathology at our university. And unfortunately, Professor Biese is recovering from the flu, so he will not be able to join us on site, but thank you for your willingness to join us online. Professor Biese.
Thank you, Mr. Chairman. First of all, my compliments with your thesis. I really loved reading it. It's really a textbook example of what private academic collaboration is capable of in the end. And of course, I also like to extend my congratulations to the supervision team. So regarding my questions, I was really intrigued by chapter 2, as well as by all the other chapters, but maybe I'll kick off with interrogating you on chapter 2. So you optimized and tested the very elegant dynamic 3-compartment model of placenta for screening purposes. And I'm impressed by this, both the setup and the outcomes that you were able to achieve. I'm a bit of a layman here, but I assume maternal endothelial arterioles directly empty into this compartment to perfuse the intervillous space. Is that correct?
Sorry.
No additional barrier between the two.
No. The endothelial cells, there's a collagen with make collagen 1 and 4, rat and human placenta, and then there are the trophoblasts. So they just there is just the ECM in between.
But is that also does that also hold for the placenta itself?
Well,
So you have a free of arteriole into the intervillous space.
we have there are the umbilical vein, then there is the ACM, and then there are like there is the trophoblast, which face that.
Okay, thanks. So I may have overlooked it, but if I look into the approaches you pursue to calculate the permeability of your model, it seems that you take a donor and the receiver compartment volumes into account, but not, or at least not what I could easily check for, the interstitial volume. Is that correct as well? What reason did you have to not include the interstitial volume into your equation?
What we do with this assay, which is an image-based assay, and we we use the intensity of the fluorescent signal that we quantify in an area that is the same from the donor and the receiver, and it's not. If we have the 3D structure like this, it's basically on the XY plane, not on the zeta plane. And this is due to the fact that we can just acquire one image, because if we, in order to consider the interface, we should have acquired multiple stacks on the zeta axis. And so that again, because that will slow down the analysis, because for each chip we should have to take multiple images, and because in one plate we have from 40 to 96 chip, for a practical matter, if we will have to take for each time point multiple zeta stack, that will we will risk that the last chips basically, if they are highly permeable, we couldn't see anything because they already been perfused. So we use when was originally developed this assay, we just we took the traditional way of using with a 2D cell culture, and then we make this simple version. And then we stick with this just one layer, because we have to take generally 12 data points over basically 12. It takes almost like 12 minutes, because to take one image per chip will take it takes a little bit of time. So that was a practical.
Now, I have another question regarding the permeability. So as you know, permeability relies on several mechanisms, depending also on the analyte that needs to permeate through the endothelium. You tested now a 70K dextrane for UVEC and 20K dextrane for CACO. This is a pretty narrow range. Did you consider to also go lower to the molecular weight of actual essential key nutrients like glucose? So in the order of 200 to 400 Dalton, or really big particles like lipoproteins that are sized 2 million Dalton up. And how would that impact on your outcome?
Thanks for your question. Well, we use in the paper, we show 70 kilodalton with the UVEC because that is the permeability, the threshold permeability of the endothelial cells, because it's considered like the dimension of albumin. And we use a smaller dimension for the gut-on-a-chip because it has a much stronger permeability. The assay, how we described in the paper and in the thesis, is meant to characterize the model in terms if it physiologically resembles the in vivo situation, and of course to check, for example, if a treatment will induce a decrease, let's say, in the barrier integrity. Of course, it could be also tested with like a compound that for example, as you said, glucose. We didn't test it, but because we visualize this assay more like for toxic study or not like for really checking, for example, the transport or like the permeability of this kind of molecule. But I think that once target the glucose will be a nice study also to, for example, in case you have a complex model, to see how it interacts, like a co-culture with this kind in this kind of context.
Okay. Thank you. If there's room for a final brief question, I'll like to go to page 56. On figure 11, you show that peroxide seems to increase the PEP, so the permeability for cell-free, but also for UVEC. But I must admit that I'm a bit disappointed by the effect size. Is it significant? And wouldn't you have expected a large effect of such a strong ROS donor on permeability?
Be sure, it's like the page where there is the trizidextran 70 kilodalton for the UVEC 54. Yes, okay, sorry. Yes, they weren't significantly different. We ran the statistic. And we could see this slightly increase in permeability just basically on the cell-free control, so on the collagen with the hydrogen peroxide. And I think that there's no significant difference, but it increases the variability. I think that this should be always included, like if some treatment are used, should be necessary to have the controls also with this treatment, especially in case of the ECM, which is a quite complex metric. So it's important to see if maybe the treatment already solubilized, for example, the collagen, and so then maybe you have this will affect also the permeability of the tubule and the stability, because if it can't, let's say, stands on the ECM because it's, let's say, full of holes, also the tubule. Thanks.
Very much for this answer. And I'll give the word back to the pro-rector.
Thank you, Professor Biesen. The opposition will now be continued by Dr. Gossijn. She is a cardiologist at our university. Unfortunately, she could not be here today because she just came out of the night shift, but thank you very much for your commitment and willingness to be here online. Dr. Gossijn.
Fantastic. Oh, I think you can hear me now, right?
Yes. We can hear you now. You can start over.
Well, thank you, Rector and dear candidate. I really would like to give you my appreciation about your thesis and the work that you provided, and also congratulate your supervising team for facilitating this, and especially for me as a medical doctor, I'm having a hard time understanding what's going on in an individual patient, and then when I look into what you guys can do and are able to, then nothing but my respect for that. It actually made my life a little bit complexer than I thought it was. And if Professor Biese calls himself a layman, I don't know what to call myself now. Yeah, obviously a lay woman, but you know what I mean. So what I was wondering about in the first place is that it is very important to, if this work can go towards what you describe it would be able to do in the near future, it would make my life much easier in prescribing drugs to pregnant women where possible. But I think what's the most important thing is here is that we keep speaking the same language. And I had a difficulty understanding major parts of your thesis, so I had to look a lot of things up. But I would like to try to make that clearer now. So I would like to start with a simple question, whether you think the placenta is a fetal or a maternal organ, because you talk about a fetal and maternal side, but what is it? Is it a fetal or a maternal organ?
Well, the placenta, esteemed opponent, thanks for your nice word and question. Well, the placenta is the interface, is considered the interface between the mother and the fetus, and they start developing few days after fertilization, so before the fetus development allows for a fetus to be developed, and it contains there's a portion of the placenta which is considered the maternal portion, and then the other one that is the fetal side. But of course, it's a kind of in between these two humans, the fetus and the mother.
So
and
does it contain?
It contains mainly the trophoblasts in different types. There are like the cytotrophoblast, in situ trophoblasts, extravillow trophoblasts, which make different parts of the placenta, and the placenta at early stage, it invades the spiral artery of the mother, and that is very important. Otherwise, we encounter preeclampsia or other different diseases. The preeclampsia is the most famous and the one who affects the majority of pregnancy. And later, and these allow also to the spiral artery to enlarge and get the perfusion necessary and the flow that we need for later stage of the development of the fetus, and to keep the nutrients and everything. So then you have the spiral artery remodeled, then you have the blood of the mother after the first trimester, and these little three that are the trophoblasts, which present the in situ trophoblasts and one layer of cytotrophoblasts. The more you go, the more it gets thinner, and then on the other side there is the umbilical vein and arteries of the fetus. So,
well, lovely, because that's exactly where I wanted to go with my second question. What you are describing is an organ in process, starting from the very first day until it gets mature at 9 months, a little bit earlier. And what I wonder is whether our understanding of the different phases of the maturation of the placenta in vivo, whether that reflects physiology, because all the only physiological placenta material that we have is at term, and all the earlier premature placentas that we may get provided are from pregnancies that got abrupted earlier due to either disease or intervenience of idiopathic factors. So how can we know that what you are developing really reflects physiology and not, in certain cases, pathology?
This is a good question. This is why also we started working in this direction, because the problem that especially at early stage is not possible to study the placenta. So we have is a little bit the chicken and the eggs, because we have to rely on 2D, on in vitro model to study an organ that we cannot study directly in human at that early stage. Of course, maybe looking for some maybe voluntary termination of pregnancy, that in that case should be still healthy, and use maybe these cells to in this kind of on a chip model, so that you have the patient material but you're still in vitro. That could be beneficial, but of course, yes, for a lot of things of the placenta we don't know, because there's not a study, or there are study based on animal, but then you're not sure. That is general things that you're never sure. Then if you go back to human, if it's the same, but the placenta is a species-specific organ, so it's a really different structure, so it's very hard. It's, I think that, of course, try as much as possible with a non-invasive technique, with in vivo, let's say, on human directly, and maybe working in parallel, like maybe a stronger collaboration with clinicians and scientists who work with this in vitro model will be very beneficial, because, of course, there's a more direct, let's say, a connection between like has that we are studying this organ, but we never, see, like, well, I saw a placenta one time when we went for a consortium in a workshop in London, but it's not something that is really familiar. Of course, this will help a lot.
Yeah, sure. Yeah. So we rely on like patients who end their pregnancy, then you mainly have the very early placental development or the very mature, and all that's in between. What I hear you saying is that we need to find some other ways to investigate that. Probably there would be a role for imaging and tissue characterization, to find some surrogates for whatever you can remodel from there. But I think it's very important to be aware of also the gaps in the development. And do I have a little bit time for another question? I don't think so. Right?
I think we have to interrupt you here.
Okay, then thank you very much for your answer.
Thank you.
Thank you, Dr. Gossijn, and the opposition will now be ended by Dr. Fouquier. He is Assistant Professor of Vascular Pharmacology at our university. Dr. Fouquier.
Thank you, Professor. Dear candidate, first I would like to address you my compliments to you and your promotion team for the nice presentation and thesis. Very interesting work, I have to say. Actually, myself as a pharmacist and as a pharmacologist, I was like pleased to read your chapter 5, and I can only acknowledge the difficulties, the pitfalls of drug discovery that you have described. And that being said, I think time for probably to discuss one point. And I would like to make a link with the initial, to close the loop, the initial discussion that Professor Sluymer has initiated on cellular heterogeneity. And actually now myself doing more fundamental research, I'm focusing on trying to understand and take into account this cellular heterogeneity as much as possible, because I realize its importance for pathophysiology. But I'm also now shared between drug screening and my roots on being a pharmacist. So I was wondering myself, is cellular heterogeneity a good or a bad thing for drug screening? And of course, I understand probably you will have to have a more nuanced answer than good or bad, but I would like to hear your arguments because that will help me a lot.
Esteemed opponent, thanks for your nice word and the question. Let's say that it depends, of course, again, heterogeneity, that means more variability, and so it goes in the opposite direction of scalability, drug development, and these kind of things. But again, we have to try to bring also complexity, because there's a problem in the early stage of drug development.
So you see this model as a kind of in-between, maybe high throughput platform and the more preclinical models.
Yes, and of course it depends what we want to resemble with the model, which kind of drug we want to develop. I think that it's highly dependent on that. The fact that we could still show that even if there were heterogeneous cells in the model, we could manage to go so high throughput. Gives a little bit of hope that it's possible, but.
Because in your chapter 5, I believe you actually screen a lot of compounds, right? A small library of compounds. But you are also not sure whether actually you are maybe missing, you maybe have also lots of false negative because some of the receptors or targets are maybe not expressed in your cells, right? Because of this heterogeneity, or maybe expressed by only a part of these cells population. So I was wondering whether how to deal with that.
I think that, well, of course, I think that is always a step by step, because if we look how generally it's done now, that sometimes are even chemical-based assay for drug development, drug discovery and screening. So there, of course, it's just the active part is very, very reductive. Yeah. Of course, in a fantastic world, we should have already like a complete heterogeneous model, like with also, for example, like the endothelial cells with also some fibroblasts or other type of cells. But before going there, we need to build a little bit all the stairs to reach that level. And I think that this is the first step. The next one is always to implement going more toward complexity together with scalability.
Actually, maybe a nice bridge with my next question, which is on your placenta-on-a-chip model and referring to more complex model, as you just said. So you show us a nice scheme of the placenta and actually vasculature in the placenta. And I was wondering if you could elaborate on the difference between the microcirculation in the placenta versus your chip model with only UVEC cells. So in terms of biomechanics, flow, diameter of channels, and so on, and how this could influence your model, how relevant it is to the human pathology.
Well, the model, of course, lack as we already mentioned, like the spiral artery. But in terms, like for example, of shear stress that we use the shear stress that we use generally for the endothelial cells, which is very close to what they should experience of the trophoblast.
Yeah, so if I recall properly, you're using this rocking platform, right, to actually move fluid across the channels. So, but is the flow actually constant, like in this technique, or is it more pulsatile in the placenta?
In the placenta, it's constant in a healthy situation.
Dear candidate Camilla Soragni, the time appointed for defending your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of a deliberation here in this room.
Ga deze kant op.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
side. 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Dear candidate Camilla Soragni, the degree committee here present has assessed the quality of your thesis and your defense. And in view of his positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Wind is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible. What is your answer to that?
Yes, I promise.
Very happy that you say that, because then we can continue to the next part, which is by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Camilla Soragni, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now, in 2 seconds, will present with you with a degree of certificate signed by the rector, the secretary, and the other members of this committee, and affixed with the official seal of the university. So, dear doctor, young doctor, very happy to be the very first one to be able to call you doctor and congratulate you. Alla tua famiglia, congratulazione con tua figlia e benvenuto in Olanda. Okay. This was really poor. So you see why I still need to continue to do this, to learn more English, Italian. But I was extremely impressed with how you presented and how you defended yourself, with coming from all different angles, the questions. But I thought it would be much more interesting, and this idea developed this week, much more interesting if the person who really knows you would give you the traditional laudatio. So this morning was a bit of a challenge, and I hope that she will also touch upon that. But we did the impossible, and I think it's much more fun to have Chi give you the classical laudatio. Maybe you can put it over there, yeah, to congratulate you. Yes, Chi.
I'm kind of told to give a speech. So, for those who don't know me, I'm Chi. I'm a senior scientist at Mimitas. I have been Camilla's supervisor at Mimitas for the last 4 to 5 years. Yeah, if I were to use one word to describe Camilla, I would say that the word is outlier, or I think in Italian it's called valore anomalo or eccezione. Yeah. So yeah, when I first met her in her interview to get the placenta scholarship, I think my first impression was that yeah, she was quite critical, quite quiet, maybe a bit bookish, but you can imagine the shock that I got when in our first company outing. I totally a very social, I don't know, party animal. But what I like her, or what attracted me to accept her as my student, was that for once, of a lot of candidates that apply, I think she's the only one that has what I call the chemistry skill sets. I thought this is something unique, something that could be an asset to the company. Yeah, and in many ways, she has really contributed a lot in that aspect. There's a lot of things that is not shown in her thesis that, yeah, she really contributed a lot to the company. And if you are at Mimitas, the first thing, or the first person that you have any questions about chemistry, you always think of Camilla. Yeah, so yeah, she's always friendly and easy to approach if you have any questions, especially like chemistry questions. Yeah, so that's maybe because of her chemistry skill sets, she's could be a considered outlier at Mimitas. But also at Mimitas, the assay development team is quite small compared to the model development team. And for us, the focus is also more on the technical aspects. So I think that's also make her quite an outlier because oftentimes she has to work quite independently, and she also has to be able to find resources, or she has to look out information by herself. I would say that, yeah, as you know, there's a pandemic, and unfortunately, due to the pandemic, I would say that if I have any regret, is the opportunity to know more about her. But having said that, in my times interacting with her, I find her to be very organized. She always likes to what I call stick to schedules, stick to a milestone. Can be quite insistent, and I think maybe one particular thing that I noticed is maybe in her writing she tends to use a bit of like Italian grammar. Yeah, the other day we were discussing the draft, and this is actually from our CEO Paul, and say, well, when I read the draft, it feels like I'm reading Italian. Yeah, but going back to the point of being an outlier, well, as you know, in science or in a lot of people, outliers can be bad or can be undesirable, especially you are in the control group or population. In statistics, people are always trying to detect you, trying to ignore outliers in their analysis, or even sometimes try to use fancy methods to remove them. But in my mind, actually, outliers can be really something good and something really useful, as you can see in her work in the screening. And what you're trying to do is actually to detect these outliers, drug candidates that in the future could be something that cure a disease or so. If anything, outliers is really, I would say, something special. And if anything, if you want to understand a system, you need to understand outliers, and you really need to pay attention to them. If anything, outliers are the one that will make your system better, and outliers complete the system. So, having said that, yeah, thank you for being part of my life journey in the last 4 to 5 years. Congratulations again on getting your PhD, Dr. Soragni. I wish you the very best in your path forward, and to hopefully you will make your own niche in the scientific field. And yeah, I hope that you will lead, excel, and overcome in your future endeavors, and do not stop to be an outlier. Thank you.
Thank you, Chi. Thank you very much.
Thank you, Dr. Ng. Dear Dr. Soragni, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. And In this congratulations, I would also like to involve your promotion team and, of course, your family, friends, and colleagues. Your promotion committee was very impressed with your performance here today. I even heard one of the better defenses we have seen lately. So I think you can put that in your bag, that very good compliment. I would also like to thank all the opponents here today, and especially thank our external opponent, Dr. Zakinia, for being here with us today. Thank you very much. And with that, I would like to close this academic ceremony. And then some household measures. I would like to invite all the family and friends already, not the family, but the friends to go to the rafter already. Family, please stay here, plus the photograph to make some pictures here, and then later on the stairs.